So after class, I guess it was maybe last Monday, one of the comments that I got back on a sticky note was it would be nice to make the assignments like ungraded homework, which I do quite a lot in this class because I want you to be working on scripting as we go. I make a number of ungraded assignments that I want you to be working on. Somebody said, oh, last Monday's was particularly involved. And so somebody made the suggestion of putting homework on the Moodle site. I'm like, oh, that's probably a good idea. So I've started doing that. And you can see from the previous week that I had done that after our meeting. What I want to call your attention to right now, though, is there are two new things up on the Moodle site. One is an Anova problem set. If you go and open this, you can see that it is basically just a set of problems. It's due the 22nd, so a week from today. There are only 4 problems, but since ANOVAs are involved, I want you to have adequate amount of time to work on them. I told you at the beginning of the semester that once you had worked out test for normality, that I wouldn't ask you to do that again. So I'm not going to have you go through and by hand check for normality. But I do want you to run a Levine's test on these data, and then I want you to run an Anova on these data. And I want you to show all your work. Now you can do this by hand if you want to, that's fine. But my advice would be to do this in an Excel spreadsheet. 2 things about an Excel spreadsheet is it just saves you some time because once you have a formula in, you can just copy that formula over and over again. The other thing that I like about an Excel spreadsheet is that when it comes time for me to grade them, I can always figure out what's going on. So let me zoom this in somewhat. So when I do this, if you're going to do it in an Excel spreadsheet, I would like you to use the tab function in Excel to show me, what, question 1 is about golf balls, question 2 is about pigs, question 3 is about GPA. And you can see that I've made just a golf balls raw data set here. That's so I could import it into R and check my work. Then I have a golf ball data set that is worked out. And I've left the structure the same. But what I've done in here is I've tried to be very careful about how I have labeled everything. So if you could help me out, be sure that if you're going to use an Excel spreadsheet, that you label everything adequately. So here's a column for sum squares total. Here is a column for the sample means. Here's a column for sum squares within. Here's a column for sum squares among. And the nice thing about this is I can then go and look at your formula and I can see if you've calculated this correctly. And then if you do me the favor of color coding things, so for example, I recalculated sum squares total over here for some inexplicable reason, but I've color coded that yellow so that I can go down here and see, well, in the Anova table, this is where sum squares total is. This is where sum squares treatment is. And this is where sum squares residual is. And so if you do that, then I know what tab applies to which question. I know what parts of your spreadsheet apply to what parts of the equations that are necessary in order to build an Anova table. What I don't want to see is just an Anova table appearing out of nowhere, because then I have no idea how you got to that Anova table, etc. And so I've already worked these out. So I have the pigs worked out. I haven't color coded it yet, and I have the GPA example worked out, but I haven't color coded it either. So if you want to do your work there, why is it not closing? That's fine. The last one of these questions is actually a question that requires you to think. And so what I've done is I've given you the sample sizes, I've given you the means, I've given you the standard deviations. And from these data, you can actually calculate an ANOVA. But in order to calculate the ANOVA, you have to go back and recall what an analysis of variance is doing. You have to recall what mean squares within is when you go to calculate this. So if you know what the standard deviation is, you know what the variance is. If you know what the variance is, then you have some idea of what within sample variation is. You know exactly what within sample variation is, et cetera. So this requires you to sit and think about what the elements of an analysis of variance are, where those elements come from, and can you extract those elements from some summary data, which you can. The next thing that is on the Moodle site is an Anova report. So I told you that there's only one exam in this class. Everything else is problem sets and reports. So in this report, I would like you to present those reports to me in PDF file, and I need to see your R work. So I need to see your R script. In the old days, before we had R Markdown, I would actually ask you for your data file and for your script. And then one of the ways that I would grade this assignment and all these other assignments is I would just load up your data file and your script, and I would run it on my own computer, and I would see if it worked and if it did the things that it's supposed to do. Now we have our Markdown, where you can basically output that report in such a way that it shows me not only what your code is, it also shows me what the output from that code is, as well as any annotations that you have is all there in a Markdown. You've seen an example of a Markdown report in the COVID data from Kansas at the beginning of the semester. So on Wednesday, what we're going to do is we're going to go through and I'm going to teach you how to use our Markdown so that you're capable of doing that. But in addition to that file, I also want a written report. So what I've done here in this file is I've given you the instructions for the report. This basically sets the context for what it's asking you to do, essentially. And then I've given you a data set that has the data in it that you're going to want to look at. And then the other thing that I've put in here is we've been working with this PEAS data set as kind of the example data set for learning how to do ANOVA. And so what I've done is I've also provided you with what I would be looking for if I was looking for a written report about the PEAS data set. So in this report, you'll see that there is a brief introduction, basically orienting the person to what the problem is, what the issue is. Why were you being brought into this as a statistician to look at these data? I do this even in instances where I've been asked to analyze somebody's data and we've had long conversations about the data before I ever sit down and analyze it, because I want the person receiving the report to be sure that I'm clear on what the questions were that they were asking and that I understand those in the same way that they understand those. And I also try and make clear what it is that I haven't been told about that data set. So for example, in this case, they were watered with these solutions and measured the growth increment after some unspecified but constant period of time. I don't know what the time period is over which these pea plants grew. If it's important for me to know it, at that point, this signals to the researcher that they need to tell me that, although it's probably not that important for what I was doing with the data. But it recognizes this is something that I don't know about this data set because this isn't my data set, it's somebody else's data set. There are a set of statistical methods, and so I talked about testing for normality and homogeneity of variances. I found that there was a significant deviation from homogeneity of variances, so then I talk about what's the transformation that I made of the data. Then I retested all of that. This is in the statistical methods because these are all things that you do before you ever go and analyze the data. And so these statistical results appear here rather than down in the results section because this is just kind of what I needed to do as a preamble to the actual analysis. And then after I've done that, I then talk about what I did, talk about the transforming and the back transformations. And then at the end, I tell the person how I did it. I did it in R. I did it in Anova in base R. There's no need to do Anova in any package because it's just one of the base functions of R. I used Levine's test from package car. I did Tukey's test using Agricola, and I did everything at an alpha level of 0.05. In this way, anybody should be able to take these data and go to the same packages I used and get the same results. And so this is something that you also find commonly in the literature nowadays. And then the results. This is the results. The results don't have to be long. They just have to tell the person what the results are. But then what you have is, in addition to this, is there are references to tables and figures in the results. Also note that when I report an F statistic in this case, I have in parentheses down here 4 and 45. That's the numerator and denominator degrees of freedom. The numerator is the degrees of freedom for treatment. The denominator is the degrees of freedom for residual. You will see that I've provided an ANOVA table, which summarizes the results of the ANOVA. You can see those 2 degrees of freedom. The reason that it's called the numerator and denominator degrees of freedom is because when you calculate an F statistic, when you calculate F, you're taking the mean squared treatment and you're dividing it by the mean square residual. Well, there are 4 degrees of freedom for treatment and there are 45 degrees of freedom for residual. That's the numerator and the denominator. It's the numerator and denominator of the F statistic calculation. So that's all there. Then there is a table that shows you the Tukey's on a significant difference results, doing all the pairwise comparisons. And then there is an informative graphic with a figure that goes with it as well. So anytime you have a table, it needs to have a caption. That caption traditionally appears above the table. Anytime you have a figure, that figure has a caption. That caption typically appears below the figure. Notice that tables and figures all have a standardized form. That form is that you have a figure number, then you have a figure title, and the title can be quite long or it can be quite short depending on the task at hand, and then you have an explanation. So in this figure, you have figure 1, differences in growth increment in pea plants watered with different sugar solutions. That's the title. That's basically telling me what this is. It's a figure with differences in growth increment in pea plants watered with different sugar concentrations. That's all the title tells you. It tells you what it is. The stuff after this tells you about those things that are in the figure. That's what the explanation is. You can see that in this figure, I actually refer back to table 2 for details on why these letters are the way they are. And it's because the control was different from everything else. Sucrose was different from control and all the simple sugars, and the simple sugars were not different from one another. And that is all found in table 2, and that is exactly what this figure shows. Control is different from everybody else. Sucrose is different from everybody else, and then these 3 things are not different from one another. That's what the letters are indicating. If you have the same letter over your mean, it means that you're not different. Different letters mean that you're different, potentially came from different populations. Same thing with this table. Here's the table number. Here is the title. What is this? Well, it's the results of the Tukey's on a significant difference test of all possible combinations of the means of pea growth. And then there's this long drawn out explanation as to, okay, that's what's in this table. What does this table mean, and how should I interpret this table? So this ended up being 1 page, 2 pages, and then bleeds over onto the 3rd page for a figure. These don't have to be long, drawn-out things, but they need to be complete. And so, because you've never done this for me before, I want you to have a template for how I'm looking for this to be done so that you kind of know what I'm expecting. So you're going to take a data set that is made up of body temperatures of lizards, and you're going to analyze it using a one-way analysis of variance, and you are going to basically write an R script, save that R script as a markdown file, which I'll teach you how to do on Wednesday, and then produce a written report. So you'll basically have 2 files, or 2 files that are combined into 1 file would be even better. One which is your written report, and then the other which is your R markdown output. Just so you can see what R markdown looks like, even though there's another example. This example exists on well, where is it? Well, I don't seem to find it. Let's go back to oh yeah, here it is. So you can, from R markdown, you can export things into a PDF, and this is the R markdown file for or the output from the R markdown file for the COVID data that we did at the beginning on masking and not masking. So here you can see that there's a block of code, and then it shows me what the output is from that code. So I did head of the data file, and then it gives me the first six rows of the data from the data file. I ask it what the date is. It's showing me that the date is character data, so I want to change that to date. So then I check it to see if that worked, and it gives me date. So it will give me all of your code, as well as all of the output from the code, including things like figures. And so in this way, I can not only look at your code and see if your code is done correctly, I can also look at your code and see if the code is giving me the proper outputs from the code. And then in some cases, it may end up being really, really long, like this one is. This will be how I'll be expecting to see things, because I can see your code, I can see your output without having to go in and run everything myself. And like I said, we'll go through Wednesday how to prepare these files. Any questions about that? All right. So hopefully you have your script that we've been working on the interminable P's data set. And so we've talked about how to run the analysis of variance. We've talked about how to create graphs of those data. We've talked about how to run Tukey's on a significant difference test. You can run that in Agricola. You can run that in CAR. It doesn't really matter. It gives you the output in slightly different ways, depending on how you're doing this. Did we run it in Agricola last time? Okay, all right, I thought we had. For some reason, that wasn't in my script. We did both. Okay, all right. I thought we had, but I don't know why it wasn't there. So You do that when you don't have planned comparisons, when you don't have an idea of what you want to compare to what. But in this case, for example, we talked about we have glucose, we have control. We have a glucose solution, we have a fructose solution, we have a glucose fructose solution that's a 1-to-1 solution, and then we have sucrose. These are all simple sugars. This is a compound sugar, and this is no sugar at all. So one of the contrasts that we might want to make is we might want to compare control to everything else. In which case there's 1, 2, 3, 4 of those, and there is one of these. And so remember that your contrast, when you're setting up contrast, they have to sum to 0. So we have these basically just set out different treatments. This is one treatment, and these are other treatments. And so it's basically what the computer is going to do is it's going to replace these factors with these things as factors. So rather than 4 factors here, you're going to have 1 factor compared to another factor in this single classification ANOVA. But you might also want to compare, for example, simple sugars to the double sugar. So since there are 3 simple sugars, you might code the simple sugar 3 and code each of these -1, -1, -1, and code this one as 0. What that tells the computer is it tells the computer to ignore this treatment altogether. I want to know, how did these simple sugars differ from the double sugar, et cetera, et cetera, et cetera. So if you have a set of planned comparisons that you want to examine before you ever go and analyze your data, there's a reason that you're running the experiment that you're running, and as a result, there are comparisons that flow out of that reasoning. You can now actually set these up beforehand, and rather than running Tukey's test, you just run planned comparisons, and you don't run Tukey's test. Now, in either case, you only run these kinds of things after you've observed a significant ANOVA. If the ANOVA is not significant, what it's telling you is that there are no differences among the means in your data set. If there are no differences among the means in your data set, it doesn't make sense to go looking for where those differences are, because there aren't any. So you only do this after a significant ANOVA, and depending on whether you had planned comparisons or you had no idea what comparisons you wanted to make, if you do see a significant ANOVA, you do Tukey's or planned comparisons. You don't do both. There's no reason to do both. If you had an a priori reason for suspecting that some things would be different from other things, then you need to test those specific planned comparisons. If you didn't have any a priori reason for thinking why things would be different, then that's when you use an unplanned comparisons method like Tukey's on a significant difference. But you don't do both. You do either one or the other, and that depends upon kind of what your knowledge of the system and what your hypotheses about that system were before you walked into doing whatever data collection you were doing. So we've done both on this data set just to illustrate to you how it gets done, but you wouldn't do this in practice. You would either do one or you would do the other. So those 2 caveats. One of the things that planned comparisons does is it gives you more statistical power to see differences, because you're not dividing your alpha level across all of these unplanned comparisons. And so that's one of the advantages of doing planned comparisons over a test that does all possible pairwise comparisons, is it limits the number of questions you can ask of your data, but as a result of that, you have more power to answer each of those questions. This brought up an interesting situation in one of my graduate statistics classes. There was a guy in our program at the school where I was getting my PhD, and when we were going through planned comparisons versus unplanned comparisons, the professor made note of the difference in power. You can see differences more easily with planned comparisons because you're limited by the overall number of degrees of freedom that you have in your experimental design. So you have more power doing planned comparisons than you do when you're doing unplanned comparisons, something like Tukey's. And the student raised his hand and said, well, can't you do Tukey's tests, and then if you find differences, you can then just go back and run planned comparisons and say that those were your planned comparisons from the beginning. And a good friend of mine turned to him and said, well, yeah, then why don't you just make up the data? So correct statistical procedure indicates that you kind of make this decision of whether you're doing a multiple comparison test versus a planned comparison test. You make that decision before you go in and start analyzing your data. You don't analyze it this way and say, oh, I found these differences. Now I'm going to call those differences planned comparisons so I can get more power to test them. You set that all up beforehand. These are sometimes referred to as post hoc tests. This just refers to after the fact. Things that you do after the fact of a significant ANOVA, you do these post hoc tests. Post hoc tests can either be planned comparisons or unplanned comparisons, but planned comparisons come from a priori hypotheses. These are before the fact. You do planned comparisons if you had some hypotheses about what your data would show before you ever went and analyzed your data. Unplanned comparisons are truly post hoc kinds of tests in that you only do unplanned comparisons after you see an ANOVA and you didn't really have any idea of what might be different from what, and so you want to explore what differences are there to see what differences are there, but you didn't have any a priori view of what would be there. So this is kind of post hoc squared, if you want to think of it that way. All right, so the question is, how do we do these planned comparisons? So the first thing that you might want to do is you might want to remind yourself of what your treatments are. And so when you do that, you're going to see we got control, glucose, fructose with a 1-to-1, and sucrose. Now this matters because as you're doing your planned comparisons, you're going to set up a series of planned comparisons, and the order in which you put the contrast coefficients matters. And so you want to know what the order of treatments are in your contrast statements. So the first thing that we need to do is so I'm going to be doing quite a lot of annotation as we go through here to keep things straight. And I always forget to put my pound sign in front. So the way we're going to do this is we're just going to say C1, use the assignment operator, and we're going to concatenate a bunch of values. 4 is for the control, and then -1, -1, -1, and -1 for the other things. So we should have 4 things in one treatment, another thing in another treatment. This corresponds to control. All of these -1s correspond to the different sugar treatments. And so when we run that, it should run without any error. And then you might want to just check yourself, just ask it to give you back C1 to see if it gives you the thing that you just gave it. So that's what the first contrast looks like. Contrast 2 might compare pure sugars to the mixture. In this case, then, C2 is going to be 0 for the first thing because you're not comparing anything to the control. But then the pure sugars are glucose, fructose, and sucrose. So those would be like maybe 1, 1, and 1. And that would mean that this could be -3. It doesn't matter which one gets the negative value and which one gets the positive value. All that matters is that they ultimately sum up to 0. And so you could have just as easily have coded this -1, -1, +3, -1, and to R it means the exact same thing. Doesn't matter. All it's basically telling you is that we have two groups. One group is the simple sugars, the other group is the mixture. One group is pure sugars, the other group is a mixture of two sugars, and it doesn't matter which ones get the negatives and positives, it's just that they need to have opposite signs. So regardless of which way you did that, you can run that. Contrast 3. Is going to be comparing among the pure sugars, and so C3 would be something along the lines of 0 for the control, so we have these other four things. So the pure sugars are glucose by itself, fructose by itself, and sucrose by itself, not the mixture. And so this is glucose, this is fructose, this is the mixture, and this is sucrose. So then we would want to have a way of telling glucose different from fructose, different from sucrose. So once again, doesn't matter which ones get the positives and negatives, except in this case, once again, you have to give them different contrast coefficients in the simple sugars, and then we're going to give the compound sugar a third contrast, and it has to be in the opposite sign because once again, contrasts always add up to 0. Now, I'm going to make a little note that this contrast consumes 2 degrees of freedom, and the reason that it consumes 2 degrees of freedom is that we have 3 different treatments that we're comparing to one another. All of the other ones had 2 treatments that we were comparing to one another. One thing versus another thing. One thing versus another thing. This thing consumes 2 degrees of freedom. So in this particular planned comparison scenario, we have contrast 1, which is control versus all the rest. This is 2 treatments effectively, which means that the degrees of freedom here is 1. Control 2 compares, or contrast 2 compares the pure sugars to the mixed sugars. Once again, pure versus mixed. But once again, because that's just 2 treatments, that's 1 degree of freedom. This third contrast compares glucose versus fructose versus sucrose. That's 3 treatments, which gives you 2 degrees of freedom. That's 4 degrees of freedom now. And you're only allowed as many contrasts as you have degrees of freedom. In your original analysis, you had 5 treatments, which gives you 4 degrees of freedom. So at this point, you're done. You've run out of degrees of freedom. So this is the limitation of planned comparisons, is it limits you to a certain number of planned comparisons that are dictated by the number of degrees of freedom in your treatment level that you have available to you. So let's try and force R to do something that it shouldn't do. Let's set up another planned comparison. Did I run that? I think I didn't run that, actually. So let's do a fourth. This will be mixed versus pure within the monosaccharides. So the monosaccharides are fructose, glucose, and we have one of those in a 2% solution, one of those in a 2% solution, and then the other is a mixed solution. So for this, the contrast, this would be C4. The contrast would be 0 for control, 1 for glucose by itself, 1 for fructose by itself, -2 for the mixture, and a 0 for sucrose. And I forgot my C, my concatenation at the beginning, which is why it's giving me that red X. So 1, 2, 3, 4, 5. And once again, that adds up to 0. And once again, it's contrasting the simple sugars by themselves versus the 2 simple sugars in a mixture. So now we've added a third contrast here, contrast 4, which is monosaccharides pure versus monosaccharides mixed, which adds another degree of freedom. We're now at 5 degrees of freedom, which is more than the degrees of freedom that we have to work with. Now, if what I tell you is true about being limited in the degrees of freedom that you have, what R should do is it should conduct this contrast, it should conduct this contrast, and it should conduct this contrast, and it should refuse to conduct this contrast. So we'll test that here in a little while. So just to test that we've got everything here, C1 is that, C2 is that, C3 is that, and C4 is that. So we have down here, we have C1, C2, C3, and C4. So we've set that all up. Now what we need to do is we need to group those things into those things. So we're going to make a matrix, and I'm just going to call it MAT because it's simple. And we want to C-bind these things together. Do you guys know what C-bind means? Stands for column bind. So whenever you see C and R at the beginning of something like this, oftentimes it refers to column and row. So what we're basically doing is we're forming a matrix, and we're forming a matrix that's going to have columns that are made up of C1, C2, C3, and C4. And then when we do that, it doesn't give us a error message. And if we type in MAT, it should give us 4 columns, column C1, column C2, column C3, and column C4. If we had done R-bind, whoa, I didn't get my R in there. I thought I had typed R. If we'd done R bind, it would give us these things in rows instead. But we don't want things in rows because our treatments are in rows, and so we want the contrast to be spread across columns, so we need to make sure that this is C bind. Once you have bound those together in a column, you now need to define those things as the contrast. So we're going to link these contrasts to the levels of the treatment. So we use the command contrast. And we want these contrasts to be spread across the treatment in our data file. And we want these contrasts to come from the matrix map that we just made. That gave us no errors there. And so then we want to view the matrix linked to these contrast elements. So we can just get dat $treatment, and it should show us that. Put your blue stickies up if you made it that far without anything self-destructing on you. Oh, you've got your green sticky. I didn't give you a sticky because you weren't here at the beginning. All right, cool. So that worked for everybody. So what we should see when we look at this is, for this first contrast, you're comparing control to everybody else. For the second contrast, you're comparing glucose, fructose, and the sucrose, the pure sugars, to a mixture of sugars. Here you're comparing among the three pure sugars. You're comparing glucose to fructose to sucrose. And so the contrast statements identify what's being contrasted with what. So let's just rerun the Anova to make sure that in all of this we haven't messed anything up. So how would we do that? What is this syntax? For running an Anova. Somebody tell me what I should type. Jonathan. We did. I know. Did you go and look at the video? Okay. Logan. AOV, all right. Oh yeah, so we're still working with the transformed data. Tilde. Okay, so let's just run this and make sure that it still runs. Hold on. Yep. What's up? Okay. Well, let's see what I get. What'd you get for your numbers?
Can you scroll back up just so I can see these? Because mine are different. There's like I've got some of my ones should be negative when they're not.
Oh, that's weird. Okay. Let me go to see where you put them in.
It's just I put them in wrong. Is what I think.
Oh, okay. All right. Yeah. Sure. So you just want to see the contrast matrix? Okay. Yeah. Sure. Right there. Is that what you need? Okay. Well, let me just go to the code. Contrast 1, there's contrast 2, contrast 3, contrast 4. Take your blue or green stickies down for now. Mm-hmm.
Says, okay, if we have like R1's positive and R3's negative.
Yeah, sure. Yeah, doesn't matter. Actually, just leave it opposite of the way that I have it, and we'll see if it gives you the same analysis. It's always nice to do things two different ways to see that what I've told you is actually correct, rather than I'm just up here making stuff up. Chance, you got it now? Okay. Yeah, so run those all over again because we're about to use them here in a minute. Okay. All right. So we've run the Anova, and it looks like it's giving us this same analysis that we got in the past. So that's good. So what we're going to do at this point is we're going to rerun the analysis, but we're going to add some things to the syntax. And what we're going to do is we're going to take all of the sums of squares of the main effect, the treatment effect, and we're going to divide that out into these 3 or 4 different contrasts. So this is a little more complicated of a list. So we've run the Anova. So it's analysis 1. So that's all we really, or I put it in as analysis 1. You could have put it in as whatever you wanted. So what we're going to do now is we're going to give us the summary again. But we're going to ask it to now split this thing up. And we're going to ask it to split it into a list of things. And this list is going to be treatment equals a sublist. And the sublist is going to be, and we're just going to give these things names. So I'm going to do control versus sugar. And this is the first contrast. Then I'm going to do mixed versus pure. Which is the second contrast. And then I'm going to do among the pure, because that is the third contrast. So what I've done is I've coded in contrast 1, contrast 2, contrast 3, and I just gave them titles so that I know what those comparisons are referring to. Now, when we run this, what it should give us is, whoa. What's that? Oh, treatment is spelled wrong. Of course it is. So I suck at typing. Thanks for that. Yeah. So what it's now showing you is that it tests the main effect at 4 degrees of freedom. It gives you the same answer that you got before. The F value has always been 53.44, and it gives you this amazingly low p-value. But then it basically takes those degrees of freedom and splits them out into one treatment that tests control versus sugar. You find that that's significantly different. You have another test that looks at mixed versus pure, and that also is significant because the sucrose pulls up the value because it has faster growth. If we go back, what we're doing in this case is we're comparing glucose, fructose, and sucrose to the mixture of the two in this contrast. And what happens is glucose is high enough that it pulls the mean of this group up enough that it makes that group different from this group, which is why it's significant, but it's not nearly as significant as the other ones. And then this treatment among pure is comparing these two, this sugar to this sugar to that sugar. And that's significant because sucrose is different from both of these. Now, one of the things that for whatever reason, and I've tried to figure this out, I ran into this problem last year, and I tried to figure out what the basis of this was. Because this is actually glucose versus fructose versus sucrose, that actually has 2 degrees of freedom. But for whatever reason, when R does the test, it tests it at 1 degree of freedom, even though we know for a fact that it's 2 degrees of freedom because we set it up. So when we get to this point, I'm asking you to do a lot of hand calculations of these tests before you get onto the computer and run them. The reason that I want you to do these things by hand is because sometimes the computer programmers who wrote this particular package that does this, they mess things up. And one of the things that they've messed up is they've messed up how the computer is treating this contrast. And they probably messed it up because usually when people are doing planned comparisons, they're comparing one thing to another thing, and then they're comparing a third thing to a fourth thing, and then they're comparing a fifth thing to a sixth thing. And so they've probably programmed it in such that the default number of degrees of freedom is 1, which seems like a stupid way of programming it, but that's the way it is. But you know, because you know what you're doing statistically, that this is not 1 degree of freedom, it's actually 2 degrees of freedom. So you need to replace 2 here so that you divide this when you go to form your mean squared so that you can then calculate the correct F value and then test that correct F value to see what its P value should be. So just because R has done it for you, no guarantee that R is doing it the correct way. The only way you know if R is doing it the correct way is by paying attention to what you're doing, paying attention to the output that R is giving you, and making sure that those things correspond. Your knowledge of what it should be doing versus what it's actually doing. So if you were reporting this, I would expect you to correct the degrees of freedom here and then do the math on this last calculation yourself and go against the output that R is giving you. Now, if this is 2 degrees of freedom, that's 2 plus 1 plus 1, that's the 4 degrees of freedom that you have available to do planned comparisons. So even though it's reporting the wrong degrees of freedom, let's see if R actually knows that it's reported the wrong degrees of freedom. Well, how can we test that? We can go back up here and we can add this last contrast, which this last contrast is, I'm going to just code it this way, G plus F versus G comma F. And this is the 4th contrast. And so this is the what was it that I did? What was MP standing for? I need to go back and look at my... It's the monosaccharides pure versus monosaccharides mixed. Monosaccharides pure versus monosaccharides mixed. If what I've said is true, and that is that this last contrast violates the assumptions of the planned comparisons, R should refuse to do this contrast. It should just dig in its heels and say, no, I'm not going to do it. So let's run this and see what we get in terms of output. It did contrast 1, it did contrast 2, it did contrast 3, and you have 0 sums of squares for contrast 4. This is basically R saying, you've exhausted your degrees of freedom. Even though I'm still only reporting 1 degree of freedom here, I R know that that's actually 2 degrees of freedom because this last degree of freedom, I'm not giving you an answer for. It gives you no sums of squares, it gives you no mean squared. I'm not sure why it gives you an F value and a P value, but basically these are zeros because you've used up all the variation that you have. The other way that you can show to yourself that you've used up all the variation that you have is by doing the math on the sums of squares. The treatment sums of squares up here is 0.6476. So for these contrasts, we have contrast 1, the sums of squares is 0.4715. For contrast 2 is 0.0356. And for contrast 3, it is 0.1406. 12 and 5 is 17. 5, 6, 7. 10, 14. 4, 5, 6. You've used up all of your variation among treatments in these first three contrasts, which means that there's 0 sums of squares left over to test contrast 4. They're off by a 10,000th of a point, rounding error. So what this does is it's basically taken whatever the variation is among treatments, and it's divided up into 3 different ways of looking at that among treatment variation. And once you've exhausted that among treatment variation, you don't have any more variation to do tests on, which is why contrast 4 returns nothing. Scientific journals have moved away from producing analysis of variance tables. Pick up a journal nowadays, and if they do an Anova, you will never see this table in the paper anywhere. You won't see it in the body of the paper. You won't see it in an appendix having to do with data analyses, et cetera, et cetera. You won't see it in the online supplemental information, which is a shame because people do all kinds of wacky stuff statistically that if you look at the Anova table, you can see where they're doing wacky stuff. Because the Anova table has an internal logic to it. Everything should add up. If it doesn't add up, something's wonky. And back in the old days when we used to publish Anova tables, just as in the body of the article that you were reading, you could look at that Anova table and see if the people were actually doing their statistics correctly. And unfortunately, because we don't tend to publish these anymore, have no idea whether you're doing stuff right or not. And so I pay attention in the body of statistics sections of papers to what their degrees of freedom are. Does that match up to kind of how they've described what their experiment is like and things like that? Because it gives me a hint as to how the Anova table should be set up. But man, I wish we could go back to the days of actually seeing an Anova table in journals, because that's the quickest and easiest way to evaluate whether or not somebody's doing correct statistical procedure or not. Because stuff like this jumps right out at you if you start adding up things like sums of squares. So, questions? What time is it? Oh, we're practically done anyway. Questions about this? Okay, so I've shown you how to do an Anova. I've shown you how to do post hoc comparisons. I've shown you how to do both planned and unplanned post hoc comparisons. Shown you how to do some graphing on how to get these numbers. Shown you the importance of back transforming from previously transformed data, because it will take graphs that look counterintuitive and turn them back into graphs that look intuitive. These are all skills that you need to know at some point in the future. On Wednesday, we'll be dealing with R Markdown. On Monday, when we come back, because we won't have class on Friday because I'm going to be out of town. On Monday, when we come back, we're going to start working on two-way analysis of variance, when you have two variables that have different levels of treatment in them. And so the section of the book that deals with that, I want you to start reading up on 2 sample tests. We're going to proceed forward with 2 sample tests, even though you're still doing homework on 1 sample test on single factor ANOVAs, simply because we need to move on. But you're going to see that a lot of the same kinds of calculations that you did in a single factor ANOVA, you're going to be doing in a 2 factor ANOVA also. It's just a little bit of a twist on things. So if anything, it'll just be reinforcing. But start on the homework as soon as you can. And then even though I'm going to be out of town, I'll still have email access. If you're having troubles with homework, you can always contact me via email, and I can help you out with that. All right, that's it for today. Put comments on your stickies, things that went well on the blue, things that went not so well on the red, and I will see you on Wednesday. Save your script. Yeah. Oh, you have a problem at the end?
No, just my numbers are like slightly off here, so it makes me think, is my treatment different? Did I use the wrong data set? Let's see, they're just like slightly. 6.408, 6.476.
Hmm.
They're just like,
They are just slightly off.
it's like, we have different, when we do summary analysis 1, which makes me think these are different when you ran yours.
Okay.
So it makes me think that my treatment's wrong.
No. You might. Have you done anything to the data file?
I mean, it's composed of, the past like 5 or 6 classes, so it very well could have been some
Yeah.
of this mess.
Go back and check and see if there's like a typo or something in the original data file.
Okay.
But that's the only thing that I can think of. Yeah. That's the only thing I can think of. Because they're all really close. That seems, whenever I see things where they're close but not exactly perfect, that's usually a sign to me that there's a typo somewhere. So on your homework, the same thing will happen. I'll see that you have values that are close but not perfect. Then I'll go and I'll probably send you an email as I grade your homework and ask you for the data file, because you're going to be inputting these things. One of the things you should always do with data input is do what we call double entry data. You enter the data into one column, then you re-enter that data again into a column, another column, and then you compare those, look for differences, and that will point out where you have typos. It's a pain. My students hate it when I make them do that. But I'm like, well, it's the only way to ensure that you've entered the data correctly. Samuel, you need to set an alarm, dude. A very loud alarm. Thanks. Yeah, you too.